Good afternoon, class. So today we're going to be talking about the nervous system. Hopefully this is review for a lot of things because we have talked about the nervous system in lab. So hopefully a lot of these terminologies and topics will be review. So the function of the nervous system. So essentially we have to gather information about our environment, all right, and that's going to be, from our skin receptors, our eyes, visions, different ways that we gather information, and it's going to all run in through the afferent, afferent, afferent pathway. That's going to be essentially, it's going to go to your brain, which is the main sensory organizer organ of the body. Okay, then your brain's going to decide what to do with that information. All right, so that's going to be the integration part. It's going to decide what to do with that information, and then it's going to send a signal out, and the signal is usually to motor neurons. A lot of times what you want to do is move your body. Okay, so usually motor output, that's efferent, efferent pathway would be the motor pathway. All right, and then you move your arm, drink your glass of water, and so forth. All right, so you can break it down into three parts. Again, the sensory input, gathering information to monitor changes occurring inside and outside the body. All right, integration is what your brain decides to do with that information, if an action is needed or not. Okay, and then the motor output. So it's going to be a response to the stimuli. Okay, and it could be activate your muscles or even glands. Like if you see food and your mouth starts to water because you're hungry. Okay, activate glands. All those things would be output. Okay. All right, so you have a central nervous system and a peripheral nervous system. So essentially, your central nervous system consists of your brain and spinal cord. All right, and then your peripheral nervous system is going to be your cranial nerves, your spinal nerves, and ganglia. And you'll see that term ganglia a lot. Ganglia are just collection of nerve cell bodies outside the central nervous system. Okay, so whenever you see the word ganglia, all ganglia are a collection of nerve cell bodies outside the central nervous system. Okay, so again, far as the functions of your central nervous system, so that's the command center. All right, it's going to interpret incoming sensory information, and then it's going to integrate that information and then send out instructions out. Okay, and it's usually your peripheral. It's going to send the instructions out usually to your peripheral nervous system, your cranial nerve, your spinal nerves. Okay, so and then those are going to send out the information. A lot of times, the motor information output to your muscles, different glands, and so forth. So the central nervous system and the peripheral nervous system, they work together. They're very dependent upon one another. All right, so the peripheral nervous system, so nerves extending from the brain to spinal cord. Your spinal nerves carry impulses to and from the spinal cord. Cranial nerves carry the impulses to and from the brain. So again, the communication lines among sensory organs, the brain, the spinal cord, and glands or muscles. So again, they're a lot of times going to be sending the signal from the central nervous system to the glands and muscles that need to be innervated. Okay, they're going to go. The sensory information is going to come in a lot of times to the central nervous system, unless it's a reflex, then it's just kind of going to the spinal cord. But unless it's not if it's not a reflex, it's going to come to the central nervous system. So the nervous system is going to decide what to do with the information. It's going to integrate it, and then it's going to send a motor signal out. okay, and it's going to send them out through the peripheral nervous system. It could either be the spinal nerve, depends on the location or what muscles need to be moved or what glands need to be activated, spinal nerve or cranial nerves, okay? So again, they work together. They're depending upon one another, um, and you can think of it as communication lines, okay, between sensory organs, the brain, and spinal cord, and glands and other muscles also. All right, so I've been using that word afferent, so sensory. The information coming in, the stimulus information coming in, um, from from your skin being touched or from what you've seen with your eyes. Okay, those are going through your afferent. All right, nerve fibers which carry information to the central nervous system. Okay, and then once it gets integrated by the central nervous system, the motor division, the efferent, efferent division is going to carry the impulse away from the central nervous system to your muscles or glands. All right, that's going to innervate those. So you should know those terminologies. Afferent, when you see that word afferent, think sensory information coming to the central nervous system. Where you see the word efferent, think of motor output going away from, carried away from the central nervous system. All right, so you have voluntary and involuntary aspects of your nervous system, just like your muscles. You have voluntary and involuntary muscles. Essentially, you have voluntary, involuntary nervous system that corresponds, that controls those muscles, essentially. Okay, so for somatic, somatic means body. So when you see the word somatic, think of your body. Okay, so somatic nervous system is going to control your skeletal muscles. Okay, and that's going to be voluntary. And then you have your autonomic nervous system, which is going to be involuntary. So it's going to be things that you don't have to think about, like your food digesting in your stomach, okay, um, and your heart beating, okay. So it's going to control smooth and cardiac muscles and glands, and they can be further divided into the sympathetic and parasympathetic nervous systems. The autonomic can be divided into the sympathetic and parasympathetic nervous system. Here's a nice diagram which kind of sums up, a nice little graphic organizer that sums up what's going on. So again, you have your sense organs, your eyes, could be your ears, hearing, could be your skin, anything that's sensory information. Because it's going to come in through the afferent pathway. All right, it's going to go up through the peripheral nervous system to the central nervous system. Now, again, if it's a reflex, all right, if it's a reflex, the sensory information is going to stop usually at the spinal cord, okay, and then it's going to send its output out, okay. But if it's not a reflex, it's going to go to the central nervous system, all right, and then it's going to, I should clarify, it's going to go all the way to the brain, okay? The brain and the spinal cord are both part of the central nervous system, but it's going to go all the way to the brain. If it's a reflex, it's just going to go to the spinal cord, and then it's going to send its output, its signal out. But if it's not a reflex, it's going to go all the way to the brain, all right? And then the brain's going to interpret the information. It's going to decide what to do with it. It's going to send an output signal, all right, out of the brain, to through the spinal cord if it's going to the lower part of the body, or it could be going to the cranial or spinal nerve. Depends on if it's going to the upper part or lower part of the body. And then it's going to go to either the somatic or autonomic um, nervous system. Okay, remember, somatic is going to be voluntary skeletal muscles. Autonomic is going to be involuntary, all right, your cardiac and smooth muscles and glands. And then your autonomic can be further broken down into sympathetic and parasympathetic nervous system. And essentially, the difference between the two is the sympathetic nervous system prepares the body for, you ever heard of that term, fight or flight? All right, responds to during any potential danger. So The sympathetic is going to be your fight or flight response, okay? And your parasympathetic, you can think of your parasympathetic, it's gonna calm everything down. All right, it's going to essentially put everything back to its normal composed state. So your parasympathetic is going to inhibit the body from overworking and essentially restore the body to a calm, composed state. Okay, so it's going to calm you down after if you got stimulated for the fight or flight, the parasympathetic is going to calm you down once that danger is essentially gone. All right, so you have support cells in the central nervous system. So let's go over some of the support cells, and we'll talk about each one of these in the slides. That's going to be coming after this one. Support cells in the central nervous system are grouped together as neuroglia. So you have different types of neuroglia, and they all function to either support, insulate, or protect the neurons. Okay, so they're support cells. You do what you think support cells are going to do. All right, they're going to support, insulate, or protect the neurons. Okay, so you have epidermal cells right here, oligodendrocytes, and this right here is satellite cells, astrocytes, microglia, and Schwann cells. And we're going to talk about each one of these different types of neuroglia in the slides coming up. So astrocytes, okay. So astrocytes have an abundant star-shaped cells. They're going to brace neurons, okay. So they're going to form a barrier between capillaries and neurons, and they're going to control the chemical environment of the brain. All right, so they're the most abundant and versatile type neuroglia. All right, so again, you see right here that the astrocytes are in between the capillaries and neurons. Okay, here's a neuron, here's the capillaries. So it forms that barrier between the capillaries. All right, capillaries and neurons. And it's also going to control the chemical environment. Okay, and that's very important. So the astrocytes are going to if you go back here, you see how they control the chemical environment of the brain. So they're going to be part of what forms the blood-brain barrier. So essentially, the blood-brain barrier is going to be formed by endothelial cells of the capillary wall, and then astrocyte infeed. Okay, the astrocyte infeed, which is going to ensheath the capillary. All right, and parasites embedding in the capillary. But you're going to hear about essentially the blood-brain barrier, and essentially, even with delivering drugs, okay, certain things don't pass the blood-brain barrier, and it's essentially to protect the brain, right? Your brain is, you can argue, the most important organ of your body, okay? So you want to protect your brain from any foreign substance, and you can have a lot of times foreign substance is in your blood, okay? So in order to protect any foreign substance in your blood from going to and essentially contaminating your brain, you have this blood-brain barrier that the astrocytes play a part of forming, okay? Also the endothelial cells of the capillary wall also help form the blood-brain barrier. But essentially certain chemicals can't get through the blood-brain barrier. And certain drugs, even if you want to take certain drugs, okay, you have to be able to, if you want it to work on the brain, you have to be able to pass that blood-brain barrier. So they will have to design drugs to be able to cross that blood-brain barrier. All right, so the microglia are spider-like phagocytes that dispose of debris. So if you remember when we talked about phagocytosis during cell structures, and you remember that phagocyte. Um, they essentially think of them as things that eat. In this case, is eats debris. Okay. Think of phagocyte. Think of a cell eating something. Okay. So the microglia are going to dispose of debris. Okay. So they're going to eat the debris essentially. Okay. Those are what your microglia cells are going to be responsible for. All right. And in your normal immune system, you have natural cells that are phagocytosis that eat foreign substances, eat foreign organisms, okay, to protect your body, essentially as part of your immune system. So these, you think of these microglial cells as defenders of the central nervous system. Think of them as your defenders of the central nervous system. Epidermal cells, okay, so they're going to line cavities of the brain, the spinal cord, and then you have cilia assist with circulation of the cerebral spinal fluid. So the epidermal cells, again, along with um, the astrocytes, okay, are going to contribute um, to the blood-brain barrier, okay. And you see these hair-like projection, this cilia, all right, which is going to help with the circulation of the cerebral spinal fluid. So your epidermal cells are going to be responsible for forming the epithelial lining of the ventricles, which are cavities in the brain, and the central canal of the spinal cord. Okay, so in these cavities, in these ventricles, cavities of the brain, and also in the um, central canal of the spinal cord. Okay, in these cavities, you're gonna have that's where you're gonna have your cerebral spinal fluid flowing. Okay, so they form the epithelial lining of those cavities, and you also in those cavities are going to have your cerebral spinal fluid flowing, and these cilia are going to help with that circulation. Okay, so again, the epidermal cells are going to form the epithelial lining of your ventricles, okay, and your central canal and your spinal cord. And again, in those ventricles and the central canal of your spinal cord, you're going to have your cerebral spinal fluid flowing, and they have these cilia that help with the circulation of the cerebral spinal fluid. All right, so your oligodendrocytes, they're going to wrap around nerve fibers in the central nervous system and produce your myelin sheaths. So in the peripheral nervous system, you're going to have a cell that produces the myelin sheath. It's going to be the Schwann cells, so don't get them confused. The oligodendrocytes, they do essentially the same thing. It's just one's located in the central nervous system and one's located in the peripheral nervous system. So in the central nervous system, it's the oligodendrocytes that's going to wrap around the nerve fibers and produce the myelin sheaths. Okay, it's going to produce these myelin sheaths essentially. And remember, these myelin sheaths are important because think of it as this way. Remember, the neurons, they have to pass action potentials, okay, down, transmit action potentials. That's how they communicate with each other, okay? So if you think of it this way, if you were trying to send a signal, okay, and if you didn't insulate the signal, it could just diffuse to the sides real easily, and it's not going to go very far, right? If you couldn't insulate it. But by insulating the nerve fibers, essentially what you're doing is you're keeping the signal from just diffusing out to the side, and then you're going to keep it going along the axon to the axon terminal. Okay, so that's what it's going to help propagate the signal, the action potential signal. That's why the myelin sheath are very important. And you even have neurological diseases that when you fail to produce these myelin sheaths, you end up with a disease state, and that's going to end up with essentially you not being able to move and death essentially. Okay, so it's very very important, these myelin sheaths. Okay, and again, don't confuse the terminology. The oligodendrocytes produce the myelin sheath in the central nervous system. Schwann cells produce the myelin sheath in the peripheral nervous system. Okay. All right, so then you have satellite cells which protect neuron cell bodies. And so you see this satellite cell, okay? This is the neuron cell body. So remember from the cell body, you're going to have the axon, okay? That's going to run off, okay? So remember, so you see the satellite cell is protecting the cell body of the neuron. So remember, I already talked about the Schwann cells. So they form the myelin sheath in the peripheral nervous system. Okay, so again, your Schwann cells are going to form the myelin sheath in the peripheral nervous system. All right, so is again still same important function. Without the myelin sheath, the action potential is not going to be able to propagate the way that it's supposed to propagate down to the axon terminal. Okay, very important. So a neuron is just a specialized cell. It's a nerve cell, okay? Specialized cell to transmit a message, okay? So we already talked about different parts of a cell. So we already talked about the nucleus, rough ER, smooth ER, mitochondria, and so forth. All right. So, but this neuron, this nerve cell, is going to be specialized to transmit signals. Okay. So it's going to need certain things that's going to enable it to transmit signals. Okay, so it's going to have a cell body. Okay, and it's going to have a nucleus, a metabolic center of the cell, and it's going to have processes. So if you look at this diagram of, well, you're also going to have Nissl bodies. Okay, those are going to be specialized rough ER, Nissl bodies, and then the neurofibrils. Those are going to be the cytoskeleton of the cell. It's going to help the cell maintain its shape and structure. And you're going to have the nucleolus. So, all right, remember we talked about the nucleolus when we talked about um, different organelles of a cell. All right, so let's go to the diagram so we can see everything. So you see these dendrocytes. So essentially the way a nerve cell works, it's got to receive a signal and then it has to send a signal out. So what's going to receive the signal is going to be the dendrites, okay? These dendrites receive the signal, okay? It's going to transmit it to the cell body. You see right here, it's the cell body. Okay, so in the cell body, it's going to decide what to do. You also see the nucleus right here in the cell body. All right, the Nissl substance. Remember, the Nissl bodies are specialized rough ER. Okay, so and you see right here mitochondria. It still has mitochondria for producing energy. So it still has organelles that we've already discussed in the cell, but it has also these specialized different structures and organelles that are specific for transmitting the signal. Okay, so the signal comes in to the dendrites, goes to the cell body, and it's going to go out through the axon. So you see right here, this is the axon hillock. So at the axon hillock right here, that's where the action potential is going to form. Okay, that's where the action potential is going to begin. All right, and then it's going to travel down the axon. Okay, so again, you see the axons covered in the myelin sheath. Okay, in this case, it's using a Schwann cell. Which, if you see a Schwann cell, then you know you're in the peripheral nervous system. Okay, and it's going to travel all the way down the axon till you get to an axon terminal. So remember, a lot of times your axon terminals are going to meet at the motor neuron junction. Okay, so at the motor neuron junction is where the axon terminal, if it's dealing with a muscle cell, is going to end. You also see in between the Schwann cells, in between this myelin sheath, you have these nodes of Ranvier. And essentially, these nodes of Ranvier are going to allow for ions to diffuse in and out of the neuron. So remember, with the action potential, ions are going to be coming in and out of the cell. Okay, so we're going to talk about action potential in the slides coming up, but essentially, sodium's going to be rushing in when you start the action potential, okay, inside of the cell. So you're gonna have to, and then you're gonna have these ions are gonna need to come in and out. So these nodes around VA allows that, okay, allows ions to diffuse in and out of the neuron, all right, propagating the electrical signal down the axon. So that's going to be important because again, you're going to need those ions coming in and out. And then when the cell is done with the action potential, when it's restoring its resting membrane potential, remember the sodium potassium pump's going to get activated, pumping 3 sodium out and 2 potassium in. Okay, it's going to restore the resting membrane potential. All right, and we're going to talk about that more in more detail coming up. So this is what I already talked about, the dendrites. They are going to conduct impulses toward the cell body. So the information coming to the cell from the afferent pathway, sensory information from the afferent pathway are going to go to the dendrites and then to the cell body, okay? And then the axon hillock is where it's going to produce the action potential, and then the axons are going to transmit the signal away from the cell body. All right. All right. So you have what's called tracts and nerves in your central nervous system and in your peripheral nervous system. So essentially, tracts are going to be bundles of nerve fibers in the central nervous system. So again, this is just terminology that mean the same thing, but based on the location, they have a different name. Okay, so don't get confused. Like we talked about the oligodendrocytes in the central nervous system, they produce the myelin sheath, and the Schwann cells do it in the peripheral nervous system. So bundles of nerve fibers in the central nervous system are called tracts. Bundles of nerve fibers in the peripheral nervous systems are called nerves. Again, it's just different terminology. Don't let that bother you or anything. And then white matter. Okay, so again, the white matter is going to be due to the myelinated fibers. Okay, when they nerve fibers are myelinated, okay, they have the white appearance. Okay, so white matter is going to be usually axon tracts. Okay, and then gray matter, since it's not doesn't have the axon tracts, it's going to be composed of essentially your cell bodies. Okay, so remember the cell bodies are not the axons. The cell bodies are the cell bodies. Okay, so the gray matter is going to be a collection of unmyelinated cell bodies. Okay, remember the cell bodies. If we go back in our image, you see right here the cell body. Okay, this is the cell body. It is not myelinated. Okay, the axon. You see this axon. This axon is myelinated. Okay, so your axon tracts are going to be essentially the white matter, and then the cell bodies, okay, what are going to be composed of what's the gray matter, what's in the gray matter, okay? So again, white matter, think of your axon tracts. Gray matter, think of your cell bodies, collection of your cell bodies, okay? White matter, collection of your axon tracts, and your axons are myelinated. Your gray matter is a collection of your cell bodies, and your cell bodies are unmyelinated, okay? You have different types of neurons in your nervous system. The multipolar neurons, it's the most common structure. So essentially the multipolar neuron has many dendrites. Okay, you see all these dendrites, so they can get a lot of information coming in. These dendrites, remember, are what allow the information coming in. Okay, so they can get a lot of information coming in, but they have one axon. Okay, so again, they have many dendrites, so that's a lot of information coming in. Okay, but they have only one single axon. Okay, and again, your polar neurons are the most common structure in the nervous system. And all your motor and your interneurons are going to be multipolar neurons. Your bipolar neurons, okay, it's going to be a type of neuron that has a cell body and two extensions. It's going to have one dendrite and one axon. And essentially, your bipolar cells are usually found in specialized sensory organs, all right, and pathways for like seeing, your eyes, smell, taste, hearing, touch, balance. All right, this is where you find your bipolar neurons. Okay, so they're usually located in special sense organs. All right, so again, they have a cell body and two extensions, one dendrite. All right, one dendrite and one axon. Okay, all right. So let's get more into nerve impulses. All right, so you have what's called a resting membrane potential. So um, essentially, the at rest, the plasma membrane is polarized. So remember, due to the sodium potassium pump, you're pumping sodium outside of the cell, and you have potassium inside the cell. But overall, um, effect of the sodium potassium pump is going to essentially result in a or around negative 90 millivolts um, resting potential on the inside, and then it's going to be positive on the outside. Okay, so if you remember from chemistry that whenever you have um, uh, a resting membrane potential like this, you have essentially generated a driving force. Okay, so because the inside of the cell is negative, around negative 90 millivolts, okay, and the outside is positive, and also you're gonna have a concentration of sodium ions on the outside. So remember when we talked about concentration gradients, particles always want to go from where they're highly concentrated to where they're low concentrated. So the combination of sodium being positive charge, all right, positive charge is going to be attracted to the negative charge, for one thing, and also the fact that sodium is concentrated on the outside, it's more concentrated on the outside than the inside. All that's going to be the driving force for sodium to rush inside of the cell once the ion gated, the voltage gated sodium channels get open. Okay, so you're gonna have these voltage gated sodium channels which are going to open, all right, when the action potential gets fired, and it's going to allow the sodium to rush inside of the cell, making it from negative to positive. Okay, positive to about plus 30, from negative 90 millivolts to about plus 30 millivolts. All right, at the height of the most positive of a action potential. Okay, so when it does that, that's called depolarizing. Okay, the cell. All right, when it's resting, it's called polarized. Okay, because you have your positive charge on the outside, negative charge on the inside. It's called polarized when it's resting. When it's action potential is going on and the sodium voltage gated sodium channels open, it's going to become depolarized, meaning that it's going to become more positive on the inside of the cell, all the way up from negative 90 millivolts to roughly positive 30 millivolts. Okay, and then it's going to end up becoming repolarized. All right, after the action potential is finished, it's going to become repolarized, and that's where the sodium potassium pump is going to help with the repolarization by kicking the sodium out. Okay, three sodium out and pumping two sodium, three sodium out and two potassium in. It's going to help repolarize the cell, essentially get the resting potential back negative. All right, so if you read the writing right here, resting membrane is polarized in the resting state. The external face of the membrane is slightly positive. The internal face is slightly negative. The chief extracellular ions is sodium, whereas the chief intracellular ion is potassium. So again, sodium is going to be concentrated on the outside, potassium concentrated on the inside. And again, you have you're gonna these ions are going to need ion channels to pass, and they're going to be relatively impermeable to both ions because they're going to essentially be closed. The ion channels for the sodium, potassium are going to essentially be closed until a voltage activates the ions to open up, allowing sodium to rush in, okay, and potassium to even leak out, okay. But again, when the cell is resting and it's polarized, the membrane is going to be pretty much impermeable to both ions. All right. All right. So depolarization. So again, depolarization is when the inside of the cell, remember it was negative, it's going to become more positive. It's going to become positive on inside of the cell. That's depolarization. So it's going to be a stimulus that polarizes the neuron membrane. What happens is you have these voltage sensitive sodium channels that are going to open up when a certain voltage gets reached, and it's going to allow the sodium to rush in, causing the inside of the cell to become more positive. Okay, so a stimulus is going to depolarize the neuron membrane. The membrane is now permeable to sodium as sodium channels open, and then the sodium is going to rush in. Okay, so let's just go over this writing. So step 2. So the stimulus initiates local depolarization. A stimulus changes the permeability of a local patch of the membrane, and sodium ions diffuse rapidly into the cell. Remember, these are voltage gated sodium ion channels. So when a certain voltage gets reached, they're going to open up, allow sodium to diffuse rapidly into the cell, and it's going to change the polarity of the membrane. So the inside is going to become more positive, and the outside is going to become more negative at that site. Okay, so the inside becomes more positive, and the outside becomes more negative at that site. All right, so action again. I talked about this in the anatomy lab, but action potentials are all or nothing, meaning you have to reach a certain threshold. If you don't reach a certain threshold, voltage threshold, if as long as you're below that voltage threshold, an active potential is not going to fire. Once you reach that threshold, the active potential is going to fire. So active potential are the all or nothing. They're either going to fire or they're not going to fire. And whether they fire or not depends on whether the threshold gets met. All right. So the movement of ions initiates an active potential in the neuron due to a stimulus. All right. So a graded potential localized depolarization exists where the inside of the membrane is more positive and the outside is less positive. So again, right here, you have a graded potential. You have this localized depolarization where the inside is more positive and the outside is less positive, more negative, essentially. So depolarization and generation of an active potential is the third step. So if the stimulus is strong enough, remember it has to reach a threshold. It's active potential to all or nothing. They're either going to fire or they're not going to fire. There's no in between. Okay. So if the stimulus is strong enough, depolarization causes membrane polarity to be completely reversed, and the active potential is initiated. All right. So propagation of the active potential. So if enough sodium enters the cell, the active potential starts and is propagated over the entire axon. So once you've reached that localized depolarization and you have enough sodium in the cell, it's going to not just be a localized depolarization, it's going to spread down the entire axon. All right, so it's no longer just a local depolarization. Once you have enough sodium, it's going to spread down the entire axon. Okay, so impulses travel faster when the fibers have a myelin sheath. So again, myelin sheaths are very important. So step number 4, propagation of the action potential. Depolarization of the first membrane patch causes permeability changes in the adjacent membrane, and the events described in step 2 are repeated. Thus, the action potential propagates rapidly along the entire length of the membrane. So remember in step 2, so essentially what's going to happen is once you have a certain voltage in this area, in that localized area, if we go back a slide, once you have a certain voltage in this localized area, it's going to open up these voltage gated sodium channels. And essentially, once the voltage gated sodium channels are open up, it's going to allow sodium to rush in. And what's going to happen is how it propagates the signal. This localized polarization is going to be spread to the next adjacent to this patch of the membrane, and it's going to open up the voltage gated sodium channels in this area. Okay, it's going to allow sodium to rush in. So it's going to keep doing that. It's going to keep allowing the voltage gated sodium channels to open up next to it, and which is going to keep the action potential propagating downward down the axon. Okay, and this is essentially step 2. I was looking at step 3, but that's step 2. Okay, and then step 2 and then 3 happens. Okay, and then it's going to continue until you propagate the action potential down the axon. All right, so you're going to have what's called repolarization, okay, of the membrane. So essentially, with repolarization, what's going to happen is you're going to restore the membrane potential back to its resting membrane potential. Okay, so you have potassium ions rushing out of the neurons after the sodium ions rush in. All right, so it's going to help repolarize the membrane. All right, so essentially repolarization is going to involve the restoring the inside of the membrane back to a negative charge and the outside back to a positive charge. Okay, so going to be different pumps that's going to help do this. Okay, so step number 5, repolarization. Potassium ions diffuse out of the cell as the membrane permeability changes again, restoring a negative charge on the inside of the membrane and a positive charge on the outside of the membrane. Repolarization occurs in the same direction as depolarization. So you're going to have these potassium channels, okay, that's going to allow the potassium to diffuse out. But you're going to also have help with the repolarization, okay, and your help's going to be through the sodium potassium pump, okay. So the sodium potassium pump is going to help also restore the membrane potential back to the resting membrane potential, okay. So you're going to use this sodium potassium pump. It's going to pump 3 sodium outside of the cell, 2 potassium in, okay. So the addition of the potassium pump that's allowing sodium with potassium channel that's allowing potassium to leak outside of the cell, in addition to the sodium potassium pump, okay, it's going to repolarize the cell, restore the cell back to the resting membrane potential, which is negative on the inside, positive on the outside of the cell. All right, so the initial, step 6, the initial ionic conditions are restored. The ionic conditions of the resting state are restored later by the activity of the sodium potassium pump. 3 sodiums are ejected for every 2 potassium ions carried back into the cell. All right, and be sure to watch this short little video on the propagation of an action potential. All right, so we talked about the action potential because that signal has to travel all the way from the axon hillock. That's where it's going to start. The generation of the action potential is going to start the axon hillock all the way down to the axon, down to the axon terminal. And remember, at this axon terminal, if you're dealing with muscle cells, you're going to end up with this neural um, neuromuscular junction. Okay, this neuromuscular junction. Okay, so and the axon terminal doesn't completely um, touch the muscle cell. You see how you have this little synaptic gap right here. Okay, so that's going to allow for um, uh, essentially a neurotransmitter, acetylcholine, to be emitted out of these vesicles of the axon terminal, and then these acetylcholine receptors, okay, are on the muscle cell are going to bind the acetylcholine is going to bind to the acetylcholine receptors, okay, which are going to result in. So let me be clear, because they similar things happen at the neuromuscular junction that happens at synapses. You can have synapses between neurons. So synapses between neurons are not the same as a synapse at a neuromuscular junction. But similar things happen. You're going to still transmit a signal through vesicles, through a synaptic cleft to another, if it's a synapse between neurons to another neuron. If it's a synapse with a muscle cell, then it's going to be to the muscle cell. So what happens at a neuromuscular junction? A synapse is very similar at the neuromuscular junction, which is the end of the axon terminal, is very similar to what happens when neurons synapse. So say you haven't reached the um, muscle, you're somewhere else in the peripheral nervous system, and your synapses in the anti-ganglia in the peripheral nervous system, or you might be synapsing there. Um, what happens is it's very similar. Okay, so again, um, if this were a neuromuscular junction, what's going to happen when action potential reaches the axon terminal? Um, Essentially, the membrane is going to increase its permeability to calcium, and calcium is going to actually help these vesicles um, to synaptic vesicles to migrate to the cell membrane and be released. All right, and these vesicles have acetylcholine. Okay, acetylcholine is going to bind to its receptors, and a lot of time the acetylcholine receptors are a ion channel that it's going to end up opening up, which is going to lead to ion permeability and the depolarization or the sarcolemma, which is the plasma membrane of a muscle cell. So that if this was a neuromuscular junction. Now, if you're synapsing, and remember the sarcolemma is going to continue into the T tubules, and the T tubules are going to pass the action potential to deep down um, into the muscle cell. But so if we're dealing with a synapse, in this case, we're talking about a synapse with another neuron. Okay. So if you are dealing with a synapses in the brain, okay, between two neurons, okay, so we're not talking about a neuromuscular junction, which is a synapse between the axon terminal and a muscle cell. We're talking about just a synapse in the nervous system between two neurons. Okay, it's going to be the similar thing as far as what's going to cause the vesicles to um, be fused with the plasma membrane and release. It's going to be the electrical charge from the action potential is going to open up calcium channels in the axon terminal, and these calcium channels are going to cause these vesicles to fuse with or I should say, allow the vesicles to fuse with the plasma membrane of the axon terminal and be exocytosed into the synaptic cleft. And when you're dealing with neurons, you have different neurotransmitters. Okay, it's not always acetylcholine. Sometimes you hear words like GABA. There are different neurotransmitters. Some are excite your nervous system. Some inhibit. Is like an inhibitory signal, propagates an inhibitory signal. So you have different neurotransmitters that get released into the synaptic cleft, okay, which is going to bind to a receptor to the synaptic cleft with the neuron. So this is going to be a neuron that's going to bind to. Okay, so I just want you to understand that when we're talking about, we talked about the signal being transferred all the way down to the neuromuscular junction, okay, and the signal can also just be transferred between neurons, okay. But this process is very similar, either one, okay. You in this, with the active potential traveling down the axon, active potential traveling down the axon, all right, to the axon terminal, and then calcium ions are going to be open due to the charge in the axon terminal, and allow that calcium, once it gets released in the axon terminal, it's going to allow the vesicle to bind with the plasma membrane and exocytose its neurotransmitter into the synaptic cleft. Okay, and the other side of the synaptic cleft is going to be a neuron if it is dealing with a synapses between two neurons. If you're not dealing with synapse between two neurons, then you're dealing with a neuromuscular junction, which is going to be a muscle cell on the other side. But again, the neurotransmitter that's released from this axon terminal, I use acetylcholine as an example because acetylcholine is one of the main neurotransmitter release in the neuromuscular junction. We are talking about um, the neuron stimulating in muscle cell, and acetylcholine will bind to a lot of times the receptor is a channel protein, an ion channel protein that's going to allow that's on the sarcolemma of the muscle cell. That's going to allow ions to diffuse into the muscle cell and then trigger a depolarization and trigger essentially another active potential in the muscle cell. Okay, and then also the sarcolemma, you have a continuation of that into the T tubule to make sure that the active potential spreads all the way in the muscle cell. But again, I'm going to go back to focusing on the neurons transmission, and it's very similar. Okay, it's just at the other side of the synaptic cleft, it isn't a muscle cell; it's going to be another neuron. Okay, that's going to be the main difference. On the other side of the synaptic cleft, it's not going to be a muscle cell; it's going to be a neuron. Okay, but you're still going to have the neurotransmitter be released into the synaptic cleft. It's going to bind to a receptor on the neuron on the other side of that synaptic cleft. Okay. All right, so this is showing you um, in more detail that calcium. Okay, so calcium in this axon terminal is going to cause these tiny vesicles containing neurotransmitter to fuse with um, the axonal membrane, the axonal terminal membrane. Okay, so calcium is going to. So again, the charge from the active potential is going to cause voltage gated calcium channels to open, allowing calcium into the axon terminal, which is going to um, cause these vesicles to fuse with the axon terminal membrane, okay, and release exocytose, its contents, into the synaptic cleft. And then you see how the receptors for this neurotransmitter is an ion channel. So essentially, before the neurotransmitter binded to its receptor, which is an ion channel, you see the ion channel is closed. Once the neurotransmitter binds to the ion channel opens up and it allows ions to diffuse into in this case, this is the other side, this is another neuron, diffuse into the neuron, triggering an active potential in this neuron, okay? That's what it's going to do. Trigger. It's going to allow this ions channel to open, which is going to allow ions to diffuse into this neuron, triggering an active potential in this neuron. So it's passing the signal along from neuron to neuron. Okay. All right. So the entry of calcium in the axon terminal causes pore-like openings to form, releasing the neurotransmitter. So again, calcium in the axon terminal is what's causing this vesicle to fuse with the plasma membrane of the axon terminal, okay, and exocytose its contents, and we call it a neurotransmitter, okay, its contents, neurotransmitter, into the synaptic cleft where it's going to bind to its receptor. The neurotransmitter is going to bind to its receptor, which is usually an ion channel, which is going to cause, when it binds to its receptor, it's going to cause that ion channel to open, which is going to allow ions to diffuse into this receiving neuron and cause depolarization there and trigger an action potential in that receiving neuron. Okay, so that's how you pass the signal on from neuron to neuron. And you're going to, again, you keep passing it on from neuron to neuron until you reach usually a neuromuscular junction. Okay, so that's why I said the process is very similar. But there is a distinction. One's a muscle cell and one's a neuron, and one's a receiving neuron. Okay, but essentially this active potential is going to go from neuron to neuron. Okay, until it reaches usually a neuromuscular junction, which is going to be the axon terminal in a muscle cell. Okay, but we're not talking about that right now. We're talking about between neurons in the nervous system right now. Okay, but again, whether this was a neuromuscular junction or between a synapse between two neurons, it's the process of this calcium, the active potential causing the calcium to ions to be released in the axon terminal, and which causes the vesicle to fuse with the axon terminal plasma membrane, causing exocytosis of the neurotransmitter. That's the same process, whether it's between a synapses between two neurons or synapses between a neuron and a muscle cell at a neuromuscular junction. It's the same process, okay? Now, if it's between neurons, these neurotransmitters might be different. Okay, it might be acetylcholine, it might be GABA. Okay, you have different neurotransmitter that your nervous system uses. Okay, some of them neurotransmitter in the nervous system when you have synapses between nerves. Some of them will be an excitatory signal. Some of them will be an inhibitory signal. Okay, so understand that there are different. These neurotransmitters can be very different between when you have synapses between neurons. Okay, you can have different neurotransmitters. Okay. All right, so the neurotransmitter molecule diffuses across the synapse and binds to receptors on the membrane of the next neuron. So again, this is a synapse between neurons, okay? So synapse between neurons in the nervous system. So you're going from one axon terminal to a of one neuron to a nerve cell on the other side, a receiving neuron, okay? And again, all you're doing is propagating the signal. And like I said, they usually keep propagating the signal until they reach a neuromuscular junction, usually what occurs, okay? All right, so if enough neurotransmitters release, greater potential will be generated. Eventually, an active potential will occur in the neuron beyond the synapse. So again, it's the same process. Active potentials are all or nothing. If enough neurotransmitter is released, you're gonna have that graded potential generated. You're gonna have that localized depolarization. Okay, and if you have enough signal, enough neurotransmitter release, and enough signal of ions rushing in, causing enough depolarization, okay, and you reach a certain threshold, then the active potential is going to fire and the neuron beyond the synapse, okay? So then the active potential is going to fire and that other neuron beyond the synapse. And again, active potentials are all or nothing. They either reach a threshold or they don't. If they don't reach the threshold, active potential doesn't fire. If you reach that threshold, active potential is going to fire, okay? All right, so the electrical change is promoted by the neurotransmitter binding up breathe. The neurotransmitter is quickly removed from the synapse. So again, there are different ways that the neurotransmitter can be broken down or removed from the synapse. Remember when we talked about the neuromuscular junction, it used the acetylcholine esterase. Okay, so it used that enzyme to remove acetylcholine. So there are different ways that the neurotransmitter is removed, but just understand that it has to be removed from the synaptic cleft. It can't stay there. If it stayed there, then it's going to constantly be activating the neuron on the other side of that synaptic cleft, and you don't want that neuron unless it is supposed to be activated to be constantly activated. So you have to remove the neurotransmitter in the synaptic cleft, okay? And there are different ways that that can occur. And you see, once that neurotransmitter it's been removed, okay, it's no longer on this receptor. You see that ion channel closes, Okay, in this case, this was a sodium ion channel because you have sodium particles that were diffusing through. But now, once that receptor, once that neurotransmitter has been removed from this ion channel, which was the receptor for the neurotransmitter, you see that no longer sodium can no longer pass through. The ion channel closes. The sodium can no longer pass through, which was causing the depolarization of the neuron of the receiving neuron. Okay. All right. So we've talked about this with the lab, but let's just go over like reflexes. So again, reflexes um, are involuntary responses to stimulus. So usually they just go from you get sensory input from your skin or wherever the input is from, and it's going to just usually go to the spinal cord. So most reflexes um, don't go all the way to the brain. So they go to the spinal cord, and then from the spinal cord, what happened? So like I was saying, um, most reflexes are involuntary, and they don't involve information being integrated in the brain. Okay, so they just go get your sensory input, and it goes to the spinal cord. Okay, then the synapse is in the spinal cord. A lot of times it synapses with an interneuron. Okay, and you may wonder, well, why do you need this interneuron if you're just going to synapse with the sensory input? It's going to just go to the output, which is a motor neuron. So, like, why have the interneuron? It's because a lot of times in this integration center, this interneuron can form another synapses with something else. So a lot of times we have reflexes. You want to activate one muscle and inhibit another. That allows one muscle to flex, and it keeps the antagonist from being activated, which will inhibit that motion from happening. So a lot of times when you do these reflexes, you are activating one set of muscles and you're inhibiting another. So these interneurons allows you to have more than one synapse. So you can have another synapse right here coming off of this interneuron to an inhibitory motor neuron. All right, that can inhibit a certain muscle from occurring because with the reflex, say I want my knee to kick out when I hit that patella tendon. Okay, I want my knee to kick out. So I want to activate um, the muscles, certain muscles in my legs, and then inhibit certain muscles um, um, in the bottom parts of my legs. So that's essentially why these interneurons are very useful. Okay, so I know when you just see the sensory neuron going to a motor neuron, you're like, why? What's the point of the interneuron? Like, you don't need the sensory. The sensory neuron could synapse directly to the motor neuron. There's no need for an interneuron if you just have that simple of a um, synapse. But a lot of times, like I said, these interneurons, the sensory can synapse with this interneuron, which can synapse with excitatory motor neuron, and then also you can have another junction on this interneuron, which can synapse with a inhibitory motor neuron, essentially. Okay. All right. So you have somatic reflexes. So reflexes that are stimulated. That stimulate the skeletal muscles. Example, you touch something that's hot, you pull your hand away. Then you have autonomic reflexes. These again, autonomic system you don't think about. So these are going to be smooth muscles, also your heart muscle and your glands. Okay, so these are things that you don't think about. Your heart beating faster if you're running or slowing down when you relax. Your blood pressure increasing, decreasing. Your glands secreting and so forth. Your food being digested in your digestive system. Okay, those are all things that can be affected by the autonomic reflexes that you don't think about, that you don't control. Okay. Or I mean, all reflexes are involuntary. So let me clarify that. So all reflexes, even somatic reflexes, are involuntary because you aren't thinking about it because they're not going to your brain, so they're not being interpreted by your brain in process. So you're not thinking about it. So it's just going to your spinal cord, and then the motor neuron is getting the output. So all reflexes are essentially involuntary, but somatic deals with skeletal muscles. So we just clarified that somatic reflexes can deal with skeletal muscles, whereas autonomic reflexes deal with smooth muscle, your heart and glands. All right, so you have five elements of a reflex. The sensory receptor reacts to a stimulus. Again, you might hit the patella tendon or touch a hot stove. Then the sensory neuron is going to carry a message to the integration center. All right, and the integration center is usually in the spinal cord. The integration center processes information and directs motor outputs. Motor neuron carries the message to an effector, and the effector organ, muscle, or gland is stimulated. All right, so this is showing you the patella tendon reflex. Stretch receptors get activated, signal goes into the spinal cord, synapses with the motor neuron, and causes then the quadricep muscles are going to contract, causing your knee to kick out. And also, what this isn't showing, which is also going on, a lot of times you have an interneuron at this synapse, all right, and then you still have the interneuron synapsing with the motor neuron that activates your quadriceps, but you also have this interneuron that's going to synapse with a another motor neuron that's going to inhibit, all right, your hamstrings from doing anything. Okay, so remember these are two antagonist muscles, the quadriceps and your hamstrings. So when your quadriceps, if your hamstrings weren't relaxed, it's not going to allow your quadriceps when they constrict. It's not going to allow that to kick your knee out. Okay, if your hamstrings aren't relaxed. So that step isn't shown there, but it shows it in some diagrams in your book. But it's just showing you the simplest pathway essentially. All right, so this is just showing you with an interneuron. So again, you got the sensory input from touch and attack. It goes in, goes to the integration center where you have your interneuron this time. So it's sensory synapses with the interneuron, which then synapses with the motor neuron. Okay, and then the motor neuron is going to end up synapsing with the effector organ, in this case, skeletal muscle. All right, so that's the end of chapter 11 notes.